So thank you everyone for coming to the BioInvestor Forum's first ever Fireside Chat with Jan Lundberg from Eli Lilly. Moderating the Fireside Chat is Simone Fishburne, Senior Editor of Cybex, BioCentury's joint publishing venture with Nature Publishing Group. Prior to joining BioCentury, Dr. Fishburne worked as a translational scientist in the industry, including 9 years of preclinical drug development at Nectar Therapeutics in their pain division. So please join me in welcoming Dr. Simone Fishburne and Jan Lundberg.
Thank you very much, and thank you everybody for coming. So I'm sure Jan needs no introduction, but I'll give one anyway. So Jan, you're Executive Vice President of Science and Technology, and as you've just told me, your preferred title is also President of Lilly Research Labs. You joined in 2010, so that's nearly 4 years there now, and your rich background includes a lot of work as an academic, experience as a biotech company founder, and obviously vast experience in big pharma and pharmaceutical industry. You're one of the most highly cited authors by the Institute of Scientific Information, publishing over 500 articles, which is many-fold what I've done, in international peer-reviewed articles. And more than that, I understand that you are not only ice hockey player in college who wanted to be a professional, but you also were recently in a tent in the snow in Alaska. Right, so we have a hardy, a hardy and worthy distinguished speaker today. So moving from Alaska to Lilly, let's talk about a little bit about the strategy for building the pipeline at Lilly. We have an audience of investors and scientists who are focused on early stage programs and drug discovery. So can you tell us about Lilly's strategy for building your early stage pipeline in particular, and how you plan to balance internal and external opportunities?
Okay, if you look at the Lilly pipeline, when I came to the company, there had already been a number of activities initiated then to improve what actually was a lack of pipeline in 2004, which had only 7 molecules in total in phase 2 and 3, which is, for a big pharma company, not very much. This has now gone to actually 39, so it is more than a 5-fold increase in molecules over these last 9 years. I had the privilege of coming into the middle of that, and you know, I could enhance some activities and I could initiate some new ones. But this is really a combination of strong internal research with then external opportunities that complement or strengthen the capabilities that we have. And just as a few examples, Lilly knew that small molecules would not be enough, and in 2004 there was only one biological project in phase 2 and phase 3 for Lilly. It was actually Byetta from Amylin. Today there is 21 programs in phase 2 and phase 3 for biologicals, and 70% of the phase 3 portfolio is actually biologicals. So a key area of strengthening was biologicals capability to then expand the opportunities to target the best target for disease, and not only targets that were amenable to small molecule drug discovery. And that was done by acquisition of some companies that had those skills, like Applied Molecular Evolution in San Diego, which was coupled then to structural genomics, which, yes, was working with small molecules, but as it turns out, structural biology is extremely helpful also to generate specific monoclonal antibodies and how to optimize them. There was also the acquisition of InClone in New York, which added additional biologicals capabilities, and the strengthening in Indianapolis. So that was a key effort. What was also done was to openly also in-license some very good molecules. One example of that is now in phase 3, baricitinib, JAK1, JAK2 inhibitor for rheumatoid arthritis with very promising phase 2 data, taking on then the Pfizer leading molecule Xeljanz in that area. Lilly also looked at, and here I also came in, we need to strengthen the diabetes area, because that was a Lilly hallmark actually since 1920. I don't know if you know that, but Lilly was the company then that worked together with Banting and Best from University of Toronto to commercialize the first insulin life-saving medicine of type 1 diabetics. So the diabetes area has been strengthened with more research, including our Shanghai site for Asian diabetes, but also then with the Boehringer Ingelheim alliance between then two pharma companies to access orals and not only have injectables as a therapy. So I think that illustrates in a way that there are a number of internal activities you can do, but you can actually synergize by doing a number of external partnerships in various ways. With academics, with biotechs, but also then with big pharmas to strengthen the portfolio. So it's not one action, it's the aggregate of action that has created this.
So, you've talked about acquisitions, and as you've built this up, as you said, building up into the biologics. As you look at acquisitions today, do you still continue to look for, would you have a preference for technologies or platforms versus individual assets, particular molecules?
What we often do with the technologies is to not acquire immediately, but first establish a partnership so we actually can test the technology and see how well it actually then can influence our own portfolio. And we have, in a number of areas, partnerships then with technology companies, and just to name a few, in the diagnostics area, which is an increasing interest from us, we have collaborations with companies like Dako or Qiagen and a number of others. In the therapeutic agent generation area, we are very keen then to look into the next generation of how you can marry small molecule with antibodies. So we have the antibody drug conjugate collaboration with Immunogen, for instance, which is moving on very well. And like I said, we looked at PET imaging as a core technology for our CNS efforts, particularly in Alzheimer's disease. And that led first to that Lilly Ventures invested in Avid Radiopharmaceuticals. And Lilly Ventures is tightly linked to the R&D organization in relation to strategy and also decisions on what they invest in. And then Avid was actually acquired and now is a core component then to look for amyloid imaging to select patients for our anti-amyloid agents, including solanezumab, which we have in phase 3, and also more recently acquiring the tau imaging ligand from Siemens, another technology platform which we see as very promising for actually looking at symptom decline in parallel then to neurodegeneration, which the tau imaging agent actually could be a much better predictor of.
So following that line then, what do you think in terms of your current portfolio and where you want to build it are the key holes? What are the, are you moving into iPS cell technologies? Are you looking at epigenetics? What are the sort of emerging areas of technologies that you think will represent important aspects for your growth?
Well, we have a strategy for R&D which we have called Timely Valued Medicines to Patients, which is an overarching philosophy in a way then to which is not rocket science, it's R&D science, and it's about coming to the patient as quickly as possible with a differentiated medicine. And we have to raise the hurdles based then on payer demands, etc. today. So we have established 5 key areas, which we call then TVM, the acronym attributes for this timely value medicine strategy, which we apply for internal programs, but also for our external in-licensing in a parallel way. And it's really about better disease understanding, having a proof of concept, preferably then before we do in-licensing, if we have an unvalidated target. we really need to tailor our medicines to the best patient. So we are looking for new diagnostic technologies, preferably then to partner with. And the third one is generating the right therapeutic agents. And I described antibody-drug conjugates as one technology. We are doing a number also of bispecific or multispecific constructs of biologicals, which we see as a key area for the future, to actually combine then the 2 for 1 or maybe 3 for 1, realizing that it will be very hard to pay for 3 expensive antibodies. It would much better to have everything in one molecule if you can do it. So that's, I think high-level criteria. Then it's always, a specific discussion then about is this asset a strategic fit or not. But like I say, we are very open to looking at opportunities, and we have a specific organization in Lilly which we call GERD, which is not a disease, gastroesophageal reflux. It's a highly, competitive global external R&D capability, and they are very keen on looking at opportunities and evaluating, and they are very much linked into our internal strategies and so on. So I would encourage you to contact Lilly, actually, if you think you have good ideas, recognizing that we have a huge influx of ideas, but I think we are also quite efficient to look at them and preferably come up then with a yes or no with a reasonable time frame.
So perhaps you can talk a little bit about how you create relationships, your relationships with venture or with VCs for your early stage programs, and when these people contact you, what it's going to look like.
Right. Well, Lilly has a VC arm itself, which I think means that we understand the needs of the VC arena quite well. And like I also said, that even if the Lilly Ventures have separate people, they're very much linked into the R&D organization in relation to strategies and future investments. So I see that as an asset. And in fact, Lilly Ventures is making profit, which I think is also great if you look at the company's overall attitude, and they have made investment into quite early stage companies. The thing we have done with VCs is probably quite unique, that we also look at portfolio assets inside Lilly, and they are presenting portfolio assets from the external environment. And we have joint activities, which we call then the capital funds, where Lilly Ventures have invested money and then working in collaboration with VCs like TVM or HCV, or also Atlas Ventures, and we are forming then small project-focused companies around single assets, which then are tested in the clinic to proof of concept in patients. And if that is possible, that Lilly has certain rights to buy these assets back and incorporate them then into the major portfolio. And we have now 8 such companies, and several of them will read out in the near future, recognizing that, yes, some of these assets are high risk, but it's also representing the breaking science with a huge upside if they work. And they're usually first-in-class molecules.
Great. So, moving in that direction, as companies approach you and they create their pitch for you, what do you think are the key hurdles? What are the key holes? What are the things that they do well, and what are the things that you think are most missing that you're looking for when a company approaches you with an asset? You talked about taking it through, phase 1. How far do they need to go? One of the questions I hear a lot is, have we done enough? Have we done too much? Should we stop yet? How do people know when they've got enough to approach?
One general experience we have is that when small companies approach us, they may say, we have a phase 3 ready asset, and a lot of the times they haven't had the regulatory interactions, and once they have that, the FDA will tell them, no guys, you need to do more studies, you haven't identified the optimal dose, what's the minimal effective dose, and what about the CMC here, and have you tested devices, etc., early enough. So I think one has to be open-minded and say that it's very important to present the idea in an objective way, because it is frustrating to hear first it's a phase 3 ready asset, and then it's probably only proof of concept in phase 2 that has been generated. So this, however, doesn't reduce our interest, in novel mechanism and novel molecules, but it's I've seen this trend over so many years that smaller companies has a tendency perhaps to be somewhat over-optimistic of the stage of their assets that they actually have generated. So that's one area which I think perhaps we can be perceived then as paternalistic, and we are just honest and open, saying that we believe that this asset probably is right here now in the development area, and by the way, we can help you to improve the asset in various ways, and then to open doors then to more realistic planning for larger, more expensive clinical trials.
So do you feel that improving that dialogue early on, helping companies like that, do you think that public-private partnerships, any of these translational forums, is the way to do that? What is Lilly's strategy for, helping those companies which ultimately might feed your pipeline to get it right?
Public-private partnerships are probably different in many ways, because as the name implies, then it's the data there are not confidential and are going to be published as quickly as possible, unless that there are some overall agreements at the starting point then about intellectual property generation and who owns what and so on. So I think one has to be more specific exactly what is the nature then of such an alliance. But what is obvious is that academia, pharma industry and biotechs and so on have formed a number of these public-private partnership over recent years. I was myself part of the group that started the Innovative Medicines Initiative in Europe, now actually a few years ago. And that, I think, is generating useful new information, but it has also taken much longer than expected. And it's some of the tasks that has been taken on there, like understanding disease and generating new biomarkers for measurements of efficacy and safety and so on, will take a long time. So I think it's important that you have realistic expectations also of the output. On the other hand, we have also seen some success stories in that area, like in the US with the dominantly inherited Alzheimer's network, the DIAN, which has followed Alzheimer's patients for a long time, looking at changes in biomarkers and symptoms in parallel, which has generated very useful information that we can use in our clinical trials then for Alzheimer agents that can potentially influence progression of disease. However, it's not really clear how venture capitalists can directly benefit from this, because I presume that what you guys want is IP protected innovations that can be exploited as new products, and that has to be done then in a way that it can actually accommodate the public part of public-private partnerships.
All right, so going back to the private partnerships, when you talk about small companies, you mentioned earlier that you sometimes sort of can advise them. Do you form sort of loose alliances? What is your strategy for bringing people to the right point where the asset is?
When we have contacts with biotech companies, it may be so that the first date doesn't end up in a marriage, right? You need to get to know the person a little bit better and have several meetings, perhaps also with some intervals, and you can actually give them advice. What do we need to see then to be really interested to buying this agent? And I think we are very open actually to telling them. Whether they want to listen or not, is another story, but sometimes it's a sequence of events that can then end up in a marriage in a way then in the end.
All right, so we'll come to some of those things, and just so everyone knows, we'll take questions shortly from the audience. But tell us a little bit. You've talked about diabetes. You've touched on CNS. Tell us about the key therapeutics areas you're in now. What you think is going to be this is the early stage. The audience, the early stage. What do you think are the key growth areas? Do you think that diabetes is one you just have to stay in the game, or do you think there's a lot of innovation there? And maybe you can focus a little on CNS as well.
Okay. Let me start with diabetes, which I know is an area actually where the VC community has been somewhat suspicious to invest in recently, based on the high hurdles from the FDA demanding cardiovascular outcomes for registration of new oral agents anyhow. On the other hand, as we all know, the diabetes is still a huge unmet medical need, and even in the US, it's only 50% of the diabetic patients, that reach their goals in hemoglobin A1C, the glucose control, and it's less than 20% that actually reach the goal in the more diabetes holistic disease, namely that people also have high blood pressure and dyslipidemia at the same time. So we at Lilly look upon diabetes as not only blood glucose control, but we want to address the other problems that these patients have, including then preventing the complications like of renal disease. So we have specific programs actually in diabetic renal disease as one interesting area, which we would like to see more innovation being done in, recognizing the huge burden then on the society for dialysis, etc., which many of these patients end up in. We have also been interested actually in the cardiovascular dyslipidemia area, since diabetics usually don't die of blood sugar problems, but they die of heart attacks, right? So, and it's still unclear exactly, how you can prevent this even by statins. So that's why we have invested in both CETP inhibitors and PCSK9. So it's more of a diabetes plus plan, recognizing, again, that yes, blood sugar is the traditional one, but there are other components that can add a lot of value. So that's diabetes. Oncology, I think we are in, like many others, then trying to tailor our medicines better, recognizing that we also have agents that recently have had positive overall survival readouts, which are broader, like ramucirumab, our VEGF-R2 antibody, which had then positive overall survival in second-line gastric cancer, both as mono and a combination therapy. But clearly we want to tailor our medicines to the population that respond best. So that's an interesting area for us. And in CNS, we are focusing on Alzheimer's disease and having a number then of approaches for the amyloid hypothesis, but also increasingly for the tau component of Alzheimer's disease, the neurofibrillary tangles. And there is some intriguing science emerging that perhaps we all have a prion disease ongoing in our brain, because the misfolded tau protein, just like then the mad cow disease protein, can spread actively throughout the brain once it has been formed. And we have some antibodies now that can prevent this, but it's another aspect of the whole Alzheimer area. And Alzheimer needs very much, earlier diagnosis, preferably before you have your dementia symptoms, because then it's probably too late. And that's why we have been so keen on getting access to imaging of amyloid and more recently also imaging of tau. So you can have that as an entry criteria and potentially also as a surrogate marker for disease progression.
So Lilly's also been historically involved in psychiatric diseases. Maybe you can talk a little bit about whether you're staying in that arena, if you think that is too high risk for you.
Well, the pharma industry usually have a dilemma, right? You're working everyone in the same area, and if it's Some people say it's too difficult. It can spread like prions also to other pharma companies, right? So we are still in the psychiatry area, recognizing though that our clinical pipeline is thinner than what it used to be, and we really need to understand the basis of psychiatric disease better. And there hasn't, been real progress, in my view, for a very long time in understanding depression pathophysiology. I mean, yes, we can inhibit uptake of monoamines in various ways, but what's the next generation of depression medicines, I think, is a key question. And here, genetics perhaps can tell us something, realizing, though, that depression, doesn't have a very big genetic component. And so I think we are very much back to the drawing board in some way here. We need to understand better the function of the brain. And some person said, to understand then the trillion of connections in the brain, it's a little bit like understanding the universe with the trillions of stars and how they interact. So that may be the problem that we have. On the other hand, academia, I think, is still very much committed, including the NIH, to do progress in psychiatric diseases based on the still then the unmet medical need and the common diseases that they have. So I think for the company or companies that stays in psychiatric disease, it will be very easy to collaborate with the best academic groups, because you're actually a single preferred partner in a way, because if all the others have left.
So you are staying in psychiatric disease?
Yeah, we are continuing in that area. Of course, we also see the very good link to Alzheimer's, because remember, the Alzheimer dementia patients also has a lot of what could be called psychiatric problems. And even the Alzheimer area need better symptomatic treatment as well, not only slowing progression of disease.
So following on the genetics line and some of the things you talked about with relation to your oncology pipeline and tailoring medicines, are you pursuing programs internally or externally around personalized medicine? Are you using patient-derived iPS cells? What are your strategies there for personalizing, for making it relevant to patients?
Personalized medicine, yes, we take advantage of the established approach of getting tumor biopsies and looking for gene mutations or overexpression then of certain pathway proteins like c-Met, for instance. We have a tailored antibody, so I think that's an obvious area. The iPS area is clearly a breaking science area. We are using that internally in our screening assays, where you can generate neurons, for instance, of various subtypes, potentially similar then to the real human neurons. At least they have a lot of characteristics that are similar. But personally, I think it's still an area for many diseases where it's too early to talk about the real impact yet. But it's an attractive technology, but it has to be better reproducible, in my view.
All right. I think. I think maybe we could take a few questions if we have some before we go on some of the things you've said. Do we have some questions? Go ahead. There's a mic behind you.
Very nice comments. How are you approaching and prioritizing data?
How we are prioritizing?
Yeah. Where are you wading into big data? How are you trying to make that something you can implement and form your programs?
Well, we generate huge amounts of data for our programs, and what we are trying to have as a principle is that we want to let patient data guide our priorities. I think we have seen over and over again, if we prioritize too early, we may be misled because the animal data may not translate to humans, and which is true both for efficacy but also for safety. We have seen so many times no animal tox, but human liver tox, for instance, or the opposite, that you have animal tox, but then there is no human tox. So patient data should guide us, I think, as a principle. Then it's always this dilemma: how much should commercial predictions guide your priorities? And this is an interesting area where, at least in my experience, the correlation between net present value judgments before you have a product and the real outcome 5 years later is very poor. Many of the blockbusters on the market have had low NPVs, even at the time of launch, because then things happen that you weren't really aware of, like competitors could fail, or suddenly then you have better results in another indication which you didn't know of, and so on. So I think you have to have a number of measures, but realizing that in the end it's still a judgment call in relation to how you prioritize, recognizing that if you have a high priority area, they have an advantage, because if you have an already established capability in sales and marketing and medical arena, of course you are much stronger to progress new agents in that particular area.
Picking up on one of those words, there have been certain drives, certainly in some of the academic things, but also in big pharma, around big data, the big words of big data, computerized approaches to gathering large amounts of data to drive your decision making, let's put it like that. Is that something Lilly is embracing?
Well, we have a very advanced capabilities in analytics that we are utilizing quite a bit in a number of areas. And remember, big data is not a new term for the pharma industry. We have been using big data in research for decades or more in how we generate our new molecules to predict new capacities of the molecules, like how do you predict toxicology or PK, or what's the rational new design to do? That's one area. Clearly, big data is in human genetics, right? Gene sequencing of individual tumor biopsies are creating huge amounts of data that needs to be interpreted, and we do a lot of analytical predictions of how we plan our clinical trials, because you can do clinical trial simulations and versus then the potential outcomes, and save a lot of efforts and time in doing that. Perhaps I should revert the question. I mean, when you talk about big data, what do you actually mean? Because it's such a broad area.
Yeah, it was intentionally an open question to see how you're prioritizing limited resources in a vast area.
Yep.
I think the exponential growth, for example, of sequencing cost, it's vastly outstripped Moore's Law, for example,
Yep.
and it surprised us. It's taken as long as it has, the 90s and into the 2000s, to make the progress we now are. But that's just a tremendous opportunity, and I know you're applying it.
Yeah.
Any more questions? Yes.
Thank you. Will you talk more about PCSK9, a fairly crowded, competitive segment?
Yeah.
A list of questions. First of all, the injectable volume, what is that? And if I remember correctly, you'll be reporting out some phase 2 data not until August of next year, or second half of 2014. How will you compete in what is developing to be a fairly crowded market? How big is that market? How do you segment it? And will a Q2-month dosing carry you? What will your delivery device look like? Is that how you're differentiating going forward?
Well, I will not be able to answer all these questions based on competitive reasons. So, but let me just summarize a few things here. the genetic validation of this target, as you know, is strong. So the likelihood that a major LDL lowering could generate then better overall cardiovascular outcome is very high. Then the question is, can you have injectable therapies in CV prevention at all, knowing that today there are generic statins, right, that do a substantial LDL reduction? So, I think it will also depend on, will the CETP inhibitors actually succeed, which, has LDL lowering some, but HDL elevation. And of course, it's really a question, well, what is best for the cardiovascular patient, including diabetics then? Is it to have a very, very low LDL, or is it to have a somewhat low LDL and a very high HDL? So and Lilly, has both PCSK9 and then CETP inhibitors in late stage clinical trials, the CETP at least. The competitiveness of this molecule, we judge then as good, although we don't have head-to-head studies in the clinic. Clearly, you can compare antibodies in other ways, and we are basing that on the data we have seen so far with our molecule. And it's we are not discussing injection volumes or devices or injection intervals, etc. currently, or maximum efficacies. But and I think it also comes out, what is then the real outcome for the front-runner molecules, Amgen and Sanofi Regeneron? Is it really that much better than a statin plus CETP, for instance, which could be a major LDL lowering and an HDL elevation, or not? And if it is a tremendous benefit, I bet there are room for more than two, and perhaps a best-in-class injectable, could be even an advantage for patients.
Do you see an injectable product being broadly used?
That remains to be seen, I think. It's clear that a more expensive injectable agent has to prove very convincing data on cardiovascular outcomes that are far better than current statins.
How do you see that developing over time? Will it take 10 years to prove out some of these theories about multi-target alternatives?
Could you repeat that?
How do you see this playing out over time? Will it take
well, we
10
will,
years to work through?
We will see within the next 5 years anyhow the various CETP inhibitors, right, and the PCSK9 antibodies, and how good data are these then agents generating in relation to cardiovascular outcome benefits. And so perhaps we should come back to this in 5 years' time when we know more.
It's a long wait, thanks.
Yeah, while, in the history of cardiovascular disease, it's not a long time. I mean, remember that. And the cardiovascular death is still the major killing reason in the world, right? And I think with the diabetes epidemic, there will still be a high need then to prevent cardiovascular outcomes. And, of course, the obesity. So.
I think we have a couple of questions from this side of the room.
For Alzheimer's, you mentioned A-beta and tau as a focus. Given the recent failures, what is your thinking on A-beta, and is there a more focused or a more differentiated view on A-beta?
Well, the A-beta hypothesis, I believe, has not yet failed. There has been some agents tested that haven't really, I think, put this on the edge in a way. I mean, there are two approaches, at least, then, that I think we should be aware of, needs to be finalized in relation to phase 3. And I'm thinking here about our own solanesumab antibody, the A-beta binding antibody that, at least in the pooled expedition and 1-2 mild patients, had a significant slowing of cognitive decline by 34%. And we have started now the next study where we are trying to reproduce that, and only including then patients that has amyloid plaques in the brain, and not everyone with dementia, which may, dilute the signal. We also have Merck's BACE inhibitor, which is a very powerful agent to lower A-beta. That, is, as I understand it, soon coming into phase 3, which is another test of the A-beta hypothesis, in addition then to some other companies' antibodies. So I think we have to wait for those data before we can say the amyloid hypothesis as influenced by pharmacology is possible. Here we realize that human genetics, I think, indicates that A-beta is likely to be important for dementia based on the hereditary early Alzheimer's disease, which accumulates A-beta then early, or the Icelandic mutation in the APP then generating less A-beta, where you can be very old and have no dementia. So if I could choose, I would like to be one of these Icelanders that had an APP mutation. I don't know about you, but it's still, I think, could be of high value. And of course, the Icelandic mutation is in the base cleavage site. So clearly the question here is if an oral BACE inhibitor actually can mimic this human mutation or not.
Can I ask one additional question? There's a dilemma. There are a lot of research programs in Alzheimer's. The best of them need to go through a proof of concept in the clinics.
Yeah.
And the clinics is very costly,
Yes.
very risky, and is often the phase 2-3 pivotal trial.
Yeah.
How do you think about that? That's something which brings biotechs onto a block.
Yeah.
And what's the responsibility, or what's the engagement of the industry? What is the engagement of the governments?
Right. It is a very important area which is discussed, I know, both by the IMI and the NIH in relation to how can we, and the FDA, which has come out with new guidelines actually of treating then dementia earlier, that how can we facilitate clinical development in the dementia area, and can we have biomarkers then that are more predictable of cognitive decline. And clearly the amyloid imaging is not really predicting decline, but more the presence or not then of a pathology. Potentially, like I said, the tau ligand, at least then in the early data, seems to be quite correlated to decline then of memory, for instance, and the MMSE judgment. So perhaps that is a better correlate. The fact remains, however, that dementia, if you diagnose early or with subtle symptoms, still in many patients is a slow progress, so it will take quite a long time before you see significant changes. Although I think we know today that if you have amyloid signal in your brain, you will actually progress faster than if you don't have amyloid in your brain with some memory problem. So that's the patient population that we now are selecting then for treatments. But it still remains a challenge being actually to really prove yourself, you need a large, long phase 3 trial.
And so, I know that in your upcoming trial, you're going for patients with mild as opposed to mild to moderate. And
yes.
Is that really just creating a huge hurdle for yourselves in terms of signal to noise, or what you'd need to see in order to get that to work?
We, like I said, we had then in the pooled mild population from Expedition 1 and 2 pre-specified statistically significant slowing of cognitive decline by 34% then versus the placebo. So we believe, like I said, by including only patients that are amyloid positive and expanding the numbers, we should be able to reproduce that and potentially have the first then disease progression slowing agent. We don't want to study moderate patients with this particular approach because we had no signal whatsoever in that patient category.
All right, I'll take some more. I think we have another question from over there.
Lee Chen here. You mentioned that you have set up an R&D center in China and then focus on the diabetes. And in the current environment, many companies try to walk away, especially from the VC world. And however, you do put more investment into this area. And then why you put a center into China? Is there a particular reason or strategy behind that, or is it disease type or differentiation of the diseases between Asian and Caucasians led you to making such a move?
Right. Well, China, as you know, is both an opportunity and a challenge. And we look upon China as a long-term opportunity where there is a huge unmet medical need in a number of diseases, including Alzheimer's. The government is putting a lot of efforts into R&D, and there is also a promise to the population for better healthcare, right? So that's a good equation. The situation also in Shanghai, to me, resembles quite a bit what happened in Boston 10, 20 years ago when all pharma companies moved there, including my old previous Astra, which was one of the first actually that moved to Boston. So pharma companies are building research capabilities in a concentrated area, particularly in Shanghai. And this together then with the talent available, including highly educated people in the US that are returning back to China with drug discovery experience, most likely tells me that there will be innovation there that we want to be part of. And we have selected then diabetes because it's one of our core strategic areas for the company, but also we feel that the problems with diabetes in Asia, including China, can be even much worse than in the US. And perhaps there is a somewhat different pathophysiology because there's not so much obesity potentially, and there are some other genetics. So for us to be on ground in China, I think has great value. And so far, we've also seen very good recruitment, of some of the best people, including some people from Merck that was involved in Januvia, are now leading our site in China. So I think we have great hopes that that will be a success story.
Great, thanks.
So that brings me just to a question that I want to ask you about Lilly and innovation and your culture. So Big Pharma have really been criticized a lot for not being fertile ground for innovation. And what are you doing, be it in China or in Indianapolis, to change that? And how are you going to get the best and the brightest to want to join Lilly rather than start their own company?
Okay. Very good question. And it's a dilemma that I've been facing over my 18 years soon then in the big pharma companies. On the other hand, there are some fundamentals that we have some great people in big pharma companies. We have in Lilly 1,500 PhDs, many that have been trained in the best American universities, Harvard, Stanford, whatever. So I think we have the people with the bright minds. It's really a question then, how do you let them innovate? And how can you encourage them to innovate? And they have also, I think, accumulated experience over the years about drug discovery and drug development that you can't really find in academia, at least. So they are quite unique. And we have some principles then that we are following that, first of all, bright people wants to be among bright people. So you need to have high levels of competence if you're going to also recruit some of the best. The other thing is that you have to give them some freedom to innovate and trust them. We talk about empowerment, and we should be very transparent, I think, in our drug discovery programs in particular, that we have to innovate then to meet the future payer demands. So there are high hurdles, which means that we will also have a high failure rate. And people should look upon that as a learning exercise, not, is that they lose their job if they fail, because then you also lose so much competence that you can use then in subsequent programs. So we are actually have a mechanism of celebrating conclusive outcomes, as we say. The important thing is to get conclusive data so that we can either progress or stop this particular approach. I know I have been quoted of saying that at Lilly we celebrate failures, which in a way we do, but in a way what I think we are celebrating is learnings. And you should also remember that people are taking on difficult tasks, and it's really about also keeping the spirit of innovation by encouraging, I think, then to move on, do the new experiment. And I can quote a very famous person that won the Second World War, more or less, Winston Churchill, and he had a lot of hurdles to overcome, and he says, well, it's easy to define success, but the way I see it, in the end you will succeed if you can go from failures to failures with enthusiasm. And that, I think, mimics a little bit the environment you have, when you're trying to invent a new medicine, because it is much more difficult than to send a man to the moon. It's probably more like to Mars, which NASA hasn't really succeeded with yet. So that's some of the aspects. On the other hand, we also require people, to constantly learn, also to be aware of what's going on externally, work with the key leading academics, publish data, be peer reviewed with your own science, and be active at conferences and so on, and talk and network then with the best academics.
And, just again, talking about risk-taking, you've talked about acquisitions, small companies that have been acquired. What are you go from a very different environment working in a small startup or biotech to working for Lilly, and how do you preserve that entrepreneurial spirit, and what are the key hurdles, or issues that you've seen with those acquisitions? And you've been on both sides of it. You've experienced both environments and an academic one.
Yeah. No, it is really the dilemma of moving, from having one idea and having to succeed with that within a limited time period, and live under this enormous pressure, and if not, you're out and there is no funding or whatever, to can you use some of that spirit then, but having a more long-term goal and test the impossible and make it possible, but be prepared of failing, because you have a long-term horizon that you can work within. And I think we have seen that with some of our programs that, like the BACE inhibitor program, for instance, it takes so long to generate molecules against some of these targets that it's unlikely that if you don't put a lot of time and effort into it, you will never succeed. So it's not an easy solution, but I believe it's really about highlighting good achievements. And you talk about entrepreneurs. I think we have within Big Pharma intrapreneurs, because we have some exceptional people that has showed over and over again that they can generate new drug discovery programs in a successful way. And those are the ones that you need to protect from bureaucracy and other interfering activities, and let them generate new programs and medicines.
Let's take a question. Oh, sorry. Go.
Yeah. I would like to talk or turn to oncology drug development and get your perspectives on how we can improve drug development success rates and also efficacy. You mentioned that you work with companion diagnostic partners like Dako and QIAGEN.
Yep.
Companion diagnostics has been around for about 15 years. I mean, the first product was launched in '98. Still, more than half of all oncology drugs fail phase 3. So far this year, there have been 19 failures in phase 3. And the efficacy is around 30%. So what do you think is needed to get better numbers in this area?
Well, we talk about, in our timely value medicine strategy, the key goal for us is not to start phase 3. The key goal is to have a successful product that the payers will reimburse so we can reach patients. And to do that, you need robust phase 2 data. So if you don't see the right things in phase 2, and can identify the right patients with a high signal, personally, I don't think you should go into phase 3 and take this much bigger bet. So I think one of the dilemmas is the rush to go into phase 3 with the wrong programs.
Go ahead.
So I'm back to the prior discussion about innovation. A number of the pharmas have set up some sort of innovation departments or groups here in the Bay Area to tap into the ecosystem. I'm wondering if Lilly has given any consideration to that.
Well, we have, West and East Coast sites. We have the Biotechnology site in San Diego, which we are expanding and have invested quite a bit in. And I know there could be some regional competition here between Bay and San Diego, but we have chosen San Diego. And then if you look at the East Coast, it's interesting because we have not chosen Boston, like every other pharma company. We have chosen New York. And actually to have then the Manhattan site, with some of the previous Implon staff. And this is a very untapped area with huge numbers of scientists and hospitals, and it's much less competition, I think, for the best minds to work with than, for instance, than at Harvard or MIT.
Any more questions? We have a clock counting down, and I'm not sure what it's counting down to be quite honest. But I think we're...
Maybe to 0.
Yeah, maybe NASA has got something going that we don't know about. But I think we're pretty much up on our hour, and it's really been a very instructive talk for us. Have you got any other lasting messages for people who may be approaching Lilly other than call us in terms of how they might deal with you, what to anticipate, timelines, patients that they need, or anything else?
No, I think when you contact big pharmas, remember that you also have to be persistent because the first contact may happen in a very unfortunate situation based on things that you don't know about and the pharma doesn't tell you. So even that if initial contact seems difficult, don't give up, but come back, and particularly if you have more data. So one interaction, is not enough in many cases.
Okay. Thank you very much.